Bismillahirrahmanirrahim. Assalamu alaikum dear students. Today we are going to start with a new chapter of the unit of renal physiology. In previous chapter we have already discussed the filtration part of urine formation, and in this chapter we are going to discuss the tubular reabsorption and tubular secretion. So as you already know that glomerular filtration is non-selective. What does this mean? This means that all the blood which is coming to the glomerulus is filtered, and all of the substances which are present in the blood are filtered into the Bowman's capsule, and this filtration is non-selective. यानी कि जो कुछ भी present है in blood, it will be filtered except your plasma proteins. Why? Because we have already discussed this. Your plasma proteins are very large in size, and also they have a negative charge on them, and as we already know that our glomerular membrane is also negatively charged, so these large plasma proteins which have negative charges on them are repelled by the negative charges of the glomerular membrane, thus they cannot filter from the glomerulus into the Bowman's capsule. So everything which is present in the blood except your RBCs and your plasma proteins, and anything that is bound to your plasma proteins such as ions and drugs, are not filtered. इनके अलावा बाकी हर चीज़ नॉन सेलेक्टिवली फिल्टर होती है. So what happens next is the reabsorption part, and the tubular reabsorption is highly selective. I have already mentioned before that your nephrons are very peculiar in their blood supply because they have dual capillary beds. When your renal artery which enters into your kidney divides into smaller branches and finally forms the efferent arterioles, which break down into the glomerular capillaries, and then they form the efferent arterioles. And suppose here you have your nephron, and your efferent arterioles then form a capillary network which is known as the peritubular capillaries, which are present all around your nephrons. So your glomerular capillaries and your peritubular capillaries are two capillary beds which are present around the nephron. In your glomerular capillaries, the forces tend to filter the fluid, and filtration is strong in your glomerular capillaries, while your peritubular capillaries are mostly responsible for reabsorption of fluid from your nephron, and they also secrete some small amounts of substances which need to be excluded from your circulation. So I hope you understand the concept of two capillary beds within your nephron. Previously I showed you a schematic diagram if you want to see how it is arranged in real life. So here you have the efferent arteriole and you have the efferent arteriole, and this is your Bowman's capsule and your proximal convoluted tubule, your loop of Henle, and your distal convoluted tubule with the collecting ducts. And these efferent arterioles, they form peritubular capillaries around your loop of Henle. Then they form your veins, and they are responsible for most of the reabsorption of substances, most filtered substances from your nephron back into your peritubular capillaries, and also a slight amount of secretion from the peritubular capillaries into the nephron. So I hope you understand this concept. So let's move on. Substances that are totally reabsorbed after being filtered in the glomerular capillaries are glucose and amino acids. So in a normal healthy person, all of the glucose and amino acids which are filtered by the glomerulus are reabsorbed in the peritubular capillaries. So none of the amino acids and glucose will appear in the urine in a healthy person. All 100% of amino acid and glucose is reabsorbed. Then there are some substances which are reabsorbed according to the needs of the body. Such substances include sodium, chloride, bicarbs, and other variable elements which are adjusted according to the needs of the body. Then there are some substances which are poorly reabsorbed and mostly excreted in the form of urine, and such substances are waste products like urea and creatinine. They are very poorly reabsorbed. Then there are various mechanisms for tubular reabsorption. Some of these require ATP and are active mechanisms, while others do not require ATP and are passive reabsorptive mechanisms. For substances to be reabsorbed, you must know the pathway of reabsorbed substances. First of all, as you already know that your nephron consists of a hollow tubular system which is made up of epithelial cells, and all of the filtrate travels in this tubular system, and the tubules are made up of epithelial cells, and around the epithelial cells are present peritubular capillaries. So anything that needs to be reabsorbed from the lumen of the nephron will have to pass first through the epithelial cells which make up the nephron, then they enter into the interstitial space, and from there they move into the peritubular capillaries and the general circulation. So this is a very important concept. You must understand this before you start studying the renal tubular reabsorption and secretion. So I have a figure for you so you can better understand. Here you can see a nephron, a part of the nephron, and as I have already mentioned that your nephron consists of Bowman's capsule and convoluted tubules, and then the loop of Henle, and then your distal convoluted tubule and your collecting duct. And this tube any part of your nephron can be zoomed in, and you can see that it is basically a hollow tube, and its walls are made up of epithelial cells, and around these epithelial cells is present your interstitial space, and adjacent to your nephron are present the peritubular capillaries. So any substance which has to be reabsorbed from the lumen of the nephron must first pass through the walls of the nephron, which are made up of epithelial cells. From there it enters the interstitial space, and from the interstitial space it enters the peritubular capillaries and the general circulation. So here is a zoomed in picture of a part of a nephron, and this is the lumen of the nephron which contains the filtrate. These are the epithelial cells which form the walls of the nephron. Here you can see the interstitial space, and here are the peritubular capillaries. So any substance which needs to be reabsorbed must pass through the epithelial cells, the interstitial space, and then reach the peritubular capillaries. Or another pathway is that it can pass between the epithelial cells to the interstitial space and then the peritubular capillaries. These epithelial cells over here have two distinct surfaces. The surface which is facing the lumen is known as the luminal surface, and the remaining surfaces are known as basolateral surfaces. This is the basal surface and this is the lateral surface, and together they are known as the basolateral surfaces. So let me repeat कि आपका नेफ्रॉन एक हॉलो ट्यूब की तरह होता है जिसके अंदर जो है लुमिन होता है, और उसकी जो वॉल्स हैं वो एपिथेलियल सेल्स से मिलकर बनी होती हैं. जो एपिथेलियल सेल्स हैं उनकी दो सरफेसेस होती हैं. एक उनकी लुमिनल सरफेस होती है जो लुमिन की तरफ होती है, और बाकी उनकी बेजोलेटरल सरफेसेस होती हैं, यानी कि बेजल सरफेस और लेटरल सरफेस. इनको इसलिए अलग से मेंशन करना ज़रूरी है क्योंकि इनके कैरेक्टरिस्टिक्स डिफरेंट होते हैं और इनके ऊपर डिफरेंट तरह के चैनल्स और ट्रांसपोर्टर्स मौजूद होते हैं. तो किसी भी सब्सटेंस को रीअब्जॉर्ब होने के लिए या तो एपिथेलियल सेल में से गुजर कर पेरिट्यूबुलर कैपिलरी में आना पड़ेगा, या एपिथेलियल सेल्स के दरमियान में से होकर इंटरस्टिशियल स्पेस में से गुजरते हुए पेरिट्यूबुलर कैपिलरीज में आना पड़ेगा. एंड कन्वर्स एंड इसके ऑपोजिट जब किसी चीज़ को सिक्रीट होना है तो उसको पेरीट्यूबुलर कैपिलरीज से निकल कर इंटरस्टिशियल स्पेस में से होते हुए एपिथेलियल सेल में से गुजर कर ल्यूमेन में आना होगा और फिर उसके बाद वो एक्सक्रीट हो सकती है in the form of urine. So as I have already mentioned that any solute that needs to be reabsorbed can pass through two pathways, either transcellular, which means through and through the cell, or the second mechanism is paracellular, which means between two cells, and this route is known as paracellular route. तो दो तरह से substances reabsorb हो सकते हैं. They can either pass through the epithelial cells or they can pass between two epithelial cells through paracellular route. Water is transported through the cells and between the cells. It uses both these pathways, the transcellular pathway and paracellular pathway. And an important, very important effect of the water is that whenever water is flowing between the cells, it carries a lot of solutes with it, and the sheer force of water helps water to carry a lot of substances along with it and help in reabsorption of these substances, and this is known as ultrafiltration or bulk flow. To explain this mechanism, let me show you a figure. So here suppose we have the lumen of a nephron, and this is the luminal side of the epithelial cells which make up the walls of the nephron, and substances can move towards the peritubular capillaries either through the cells or between the cells. The pathway which is through the cells is known as transcellular pathway, and the pathway which passes between the cells is known as paracellular pathway. Water can also be transported by both these routes, and whenever there is a lot of solute present in the interstitial space, it is going to attract a lot of water from the lumen into the interstitial space, and water is going to move through both these pathways and get reabsorbed into the blood, and it is going to take a lot of solutes with it, and this is known as ultrafiltration or bulk flow. ठीक है, तो जब सॉल्यूट्स बहुत ज़्यादा जमा हो जाते हैं इंटरस्टिशियल स्पेस में तो वो वाटर की रीएब्जॉर्प्शन को प्रॉम्प्ट करते हैं, और वाटर ल्यूमेन से मूव करता है टूवर्ड्स द इंटरस्टिशियल स्पेस एंड देन इनटू द पेरिट्यूबुलर कैपिलरीज, और वो अपने साथ इन सॉल्यूट्स को भी बहा कर ले जाता है, एंड दिस प्रोसेस इज़ नोन एज बल्क फ्लो or ultrafiltration. This is very important from the point of view of MCQs. Next, let us discuss the mechanism of active transport. You already know that whenever there is use of ATP during transportation, this is known as active transport, and it is always uphill, which means it is always against the concentration gradient from low concentration to higher concentration of substances. That is the reason it requires energy and ATP. So let us discuss how the active transport takes place and what are the substances which are transported through active transport. So as I already mentioned that active transport is always against the electrochemical gradient, and energy is utilized by hydrolysis of ATP, and examples are the sodium potassium ATPase pump, the hydrogen ATPase pump, hydrogen potassium ATPase pump, and calcium ATPase pump. These pumps are present in specific parts of the nephron. For example, the sodium potassium ATPase pump is present almost along the whole length of nephron, but it is very important in the principal cells of distal convoluted tubule. Then we have hydrogen ATPase pump, which is present in the collecting ducts, we have hydrogen potassium ATPase pump, which is present in the intercalated cells of the distal convoluted tubule, and so on. We will be discussing different types of transporters when we will discuss different parts of the nephron. अभी आपको सिर्फ ये पता होना चाहिए कि एक्टिव ट्रांसपोर्ट की एग्जांपल्स जो हैं they are the sodium potassium ATPase pump and other pumps. And as you can see that this is all primary active transport. Why? Because primary active transport is the type of transport in which direct ATP is cleaved, and you can see ATP being broken down by the ATPase protein. Then here we have an example of sodium potassium ATPase pump, which is present in the basolateral surfaces of the epithelial cells, and they are involved in the reabsorption of sodium. So when most of the sodium is filtered at the Bowman's capsule, while it is passing through the proximal convoluted tubule, almost 65% of it is reabsorbed. So how this reabsorption takes place? First, you must remember that sodium is an extracellular ion, and its concentration is very high outside in the interstitial fluid and in the peritubular capillaries. Its concentration is low within the cells. So when sodium is passing from the lumen of the nephron, it is reabsorbed by facilitated diffusion from the lumen into the cell along its concentration gradient. But as the time passes, sodium is going to start accumulating in the cell and the gradient is going to disappear. Then no further sodium can be reabsorbed in the cell, and this may lead to excretion of sodium in the urine or loss of sodium in the urine. We cannot allow this to happen. So to maintain a gradient, there are present sodium potassium ATPase pumps at the lateral and basal surfaces of the epithelial cells. Now what they do is they pump 3 sodiums out of the cell into the interstitial space and pump 2 potassium into the cell. When the sodium is continuously being pumped out from the cell, it reduces the concentration of sodium within the cell, so more sodium can be reabsorbed from the lumen into the cell. ठीक है, तो बेटे ये एक सिंपल कॉन्सेप्ट है कि सोडियम जब ल्यूमिन में से मूव कर रहा होता है तो उसको रीअब्जॉर्ब करने के लिए यहाँ पर कुछ प्रोटीन्स मौजूद होती हैं, और ये फैसिलिटेटेड डिफ्यूजन से सोडियम जो है वो रीअब्जॉर्ब होता है या इट इज़ transported from the lumen into the cell. But as sodium starts to accumulate in the cell, further transport of sodium from the lumen into the cell will be reduced. तो इस सोडियम ग्रेडियंट को मेंटेन करने के लिए सोडियम को एक्टिवली पंप आउट किया जाता है फ्रॉम द सेल इनटू द इंटरस्टिशियल स्पेस विद द हेल्प ऑफ सोडियम पोटेशियम ATPase pump which is present at the basal and lateral surfaces. And as you already know, there is a lot of sodium in the extracellular fluid, that is your interstitial space and your peritubular capillaries. So to pump sodium from low concentration into a higher concentration environment, you are going to need ATP, and this ATP is cleaved and sodium is pumped out against its concentration gradient to maintain a low concentration of sodium in the cell, so more sodium can be reabsorbed from the lumen. This is how sodium is reabsorbed from the lumen into the peritubular capillaries. And as sodium accumulates in the interstitial space, it is going to prompt the reabsorption of water from the lumen into the interstitial space, and this water is going to take a lot of sodium with it and enter into the peritubular capillaries, as I already mentioned in the previous slide, and this is known as bulk flow or ultrafiltration. Next we have the mechanism of secondary active transport. In secondary active transport, no direct ATP is being cleaved, but the transport of substances is taking place against the concentration gradient, and this gradient is maintained in the background by sodium potassium ATPase pump. So let me clear your concept about secondary active transport. What happens in secondary active transport is that a substance which is moving along its concentration gradient takes along with it a substance against its concentration gradient. So suppose we have sodium which is present in greater quantities within your lumen, but sodium in your cell is very little in concentration, so the sodium is going to move from the lumen into the cell. This gradient is so huge that a sodium can also take some other substance along with it against its concentration gradient. Suppose there is present glucose in the lumen, and glucose is in higher concentrations within the cell as compared to the lumen, and normally glucose would not move from the lumen into the cell, but sodium which is moving along its concentration gradient creates such a huge gradient and has so much energy that it can take glucose along with it against its concentration gradient. So this is known as secondary active transport. Here you can see no ATP is being cleaved by this protein, so there is no direct breakdown of ATP, but as I mentioned in the previous slide, that sodium can only move into the cell as long as the concentration of sodium in the cell is low. लेकिन जो cells के अंदर sodium आता जाएगा, the calcium the concentration of sodium is going to increase, and a time is going to come when no more further sodium can enter the cell, and neither can it bring glucose along with it. So at this point, the sodium potassium ATPase pump, which is present at the basal and the lateral surfaces, are going to throw this sodium out so that the sodium concentration within the cell can be reduced and more sodium can enter into the cell and bring along glucose. So there is no direct breakdown of ATP, but in the background the energy E is being provided by the sodium potassium ATPase pump in the form of sodium gradient. So let me explain this in the form of a definition, that whenever there is movement of substances against their concentration gradient, but there is no direct cleavage of ATP, instead the energy is being provided in the form of sodium gradient. This is known as secondary active transport. So two or more substances are transported together with the help of a carrier protein, and these substances can both travel in one direction or they can be traveling in an opposite direction. When the direction is same, this is known as cotransport or symport, and when the direction is opposite, this is known as counter transport or antiport. So we will see an example of both of these in the next slides. When one of the substances, for example sodium, diffuses along its concentration gradient, the energy released is used to drive another substance, for example glucose, against its concentration gradient, and this is known as secondary active transport. And sodium का gradient कौन maintain कर रहा है? ये sodium जो सेल के अंदर जब आएगा और सेल के अंदर accumulate होना शुरू हो जाएगा, तो इसके सोडियम को continuously बाहर कौन निकाल रहा है ताकि एक gradient हमेशा के लिए बना रहे? That is sodium potassium ATPase pump. So there is no direct usage of ATP, but there is indirect ATP energy in the form of sodium gradient, which helps sodium to enter the cell and bring along another substance against its concentration gradient. So this is known as secondary active transport. and there is no direct usage of ATP. Secondary active transport can be useful in the form of reabsorption and also in the form of secretion. Now reabsorption is when both of the substances are moving into the cell, and the example is sodium taking along glucose with it, and sodium taking along amino acids with it. So sodium glucose and sodium amino acid are an example of co-transport and example of active reabsorption. But in some other instances there are such proteins present which attach sodium on the outer side, and they attach another substance from inside the cell and they throw it outside against its concentration gradient, and here you can see that this will result in the secretion of this second component. And such examples are sodium hydrogen exchanger and sodium potassium exchanger. So either sodium can move in and throw out a hydrogen ion, or sodium can move in and throw out a potassium ion, and these are known as sodium hydrogen and sodium potassium exchangers, and this is an example of antiport or counter transport. Here are some figures to better understand the secondary active reabsorption, and there are two examples of glucose and of amino acids. So here we have this figure in which this is the luminal surface and this is the basal surface, and here you have the peritubular capillaries. So what happens is, along the luminal surface the sodium moves from higher concentration to lower concentration within the cell, and it also takes along glucose against the concentration gradient of glucose, and the proteins over here do not use any ATP, but this sodium which is accumulating within the cell is being continuously pumped out by the sodium potassium ATPase pump, which generates a gradient within the cell so that more sodium can move inside. And the proteins which are involved are the sodium glucose cotransporter. Here this protein is known as sodium glucose cotransporter, and once when glucose accumulates within the cell, it simply diffuses out through glucose transporters, which are not energy dependent and they are passive transport channels, and glucose can simply leak out of the cell along its concentration gradient. So another example is sodium amino acid cotransport. Sodium, when it enters the cell, it brings along amino acids with it, and then this sodium is thrown out with the help of a sodium potassium pump, and amino acids diffuse out of the cell into the interstitial space and then into the peritubular capillaries. So this is an example of secondary active reabsorption. Here we have an example of secondary active secretion, and here we have an anti-transport or counter transport. So sodium enters into the cell along its concentration gradient, and this concentration gradient is being provided by the sodium potassium ATPase pump, and it pushes out potassium against its concentration gradient, and you can see that the direction is opposite, so this is known as an antiport or counter transport. And since hydrogen is being secreted, so this is an example of secondary active secretion, and this protein over here is known as a sodium hydrogen exchanger, and you can see that no ATP is being broken over here, so this is not an example of primary active transport. In fact, it is an example of secondary active transport. Then for larger sized substances, there is a special mechanism for reabsorption or transport. This is known as pinocytosis, in which the epithelial cells invaginate and take in substances along with some fluid, and this is pinched off and enter the cell in the form of a vesicle. We have already studied what is meant by pinocytosis in the first unit of cell, so I don't think we need to revise this, but remember that pinocytosis also occurs at the luminal surface of epithelial cells of the nephron to absorb larger sized substances. Now, if you remember from your unit of cell that any substance which is being actively transported through a carrier protein reaches a specific threshold beyond which it cannot be reabsorbed, and this is known as transport maximum. There is a simple reason for this. Suppose this is your Bowman's capsule, and within the walls of your Bowman's capsule you have specific carrier proteins which are present at the luminal surface of the cells. So you have them present over here and on both sides, and they are responsible to transport a certain type of substance from the lumen into the cell and into the interstitium. So suppose this is a carrier protein, and the substance which is being filtered from the glomerulus enters into the Bowman's capsule, and as it passes down the tubular system, it is being reabsorbed by these carrier proteins. And if the carrier proteins are not saturated, they are going to absorb all of the substance. But if the filtration of that substance increases to such an extent that all of these carrier proteins become saturated, so much so that they cannot reabsorb all of the substance, this substance is now going to be excreted in the urine, and it is going to start appearing in the urine. And the reason for this is that its filtration is greater than the capacity of the carrier proteins to reabsorb it, and it exceeds the number of carrier proteins. So this is known as the transport maximum of that substance, because it is the carrier proteins have been saturated and no more substance can be reabsorbed. So transport maximum is the limit to the rate at which a solute can be transported, and it is due to the saturation of the carrier proteins when the tubular load, which means the solute which is filtered in the tube per minute, exceeds the capacity of the carrier proteins. So this is very logical that when the carrier proteins, which are only present in a specific number and work at a specific rate, when they are overwhelmed by greater than normal secretion of a substance, of course they are going to reach a maximum and a threshold beyond which they cannot reabsorb that substance, and now this substance is going to appear in the urine and is going to be excreted. This is known as the transport maximum of that substance, and this happens when the filtered load. What is meant by filtered load? It means the amount of substance which is being filtered per minute, and when this filtered load increases, exceeds the capacity of the carrier proteins present within the lumen of the nephron, this is known as the transport maximum of that substance. Normally transport maximum is seen for the glucose in conditions such as diabetes mellitus. In a healthy person, no glucose appears within the urine, and all of it is reabsorbed by the carrier proteins present in the epithelial cells of the nephron. But in conditions when there is so much glucose which is being filtered by the glomerulus into the Bowman's capsule that it exceeds the capacity of the carrier proteins to reabsorb this glucose, this is known as the tubular maximum of glucose, and it does not happen in a normal healthy person. This only happens in people suffering from diabetes mellitus, because in such people there is absence or insensitivity to insulin, and when insulin is not present, the glucose in your blood cannot enter in the muscles and in different cells of your body, and its concentration in your blood is greatly increased, and it is going to be filtered in great quantities from the glomerulus into the Bowman's capsule. And when this filtration exceeds a threshold, the glucose starts appearing in the urine. This is known as transport maximum of glucose. So there is a graph to better understand the transport maximum of glucose. To understand this graph, first you need to understand three terminologies. The normal tubular load of glucose is around 125 milligrams per minute. So the amount of glucose which is being filtered from the glomerulus into the Bowman's capsule in a normal healthy person is around 125 milligrams per minute, and in a normal healthy person all of this is reabsorbed. But in a person who is suffering from diabetes mellitus, this tubular load exceeds 125 milligrams, and by the time it increases to 200 milligrams per minute, some glucose starts to appear in the urine, and this is known as the threshold for the secretion of or excretion of glucose in the urine. And when further glucose concentration in your blood increases, there comes a time when all of this glucose is now going to appear in the urine, and this concentration at which that the glucose starts appearing in the urine is known as transport maximum, and it is around 375 milligrams per minute. So let me explain this with the help of a graph. I hope you understood these three terminologies. Now let us put these three terminologies to a practical graphical representation. So in this graph we have plasma concentration of glucose on the x-axis. Here we have the plasma concentration of glucose, and on the y-axis we have the amount of glucose which is being filtered, reabsorbed and excreted. So you know that as the no matter how high the concentration of glucose is in your plasma, all of it is going to be filtered into your Bowman's capsule. So the graph of filtration is always a steep straight line. Why? Because I mentioned before that everything that is present in your glomerulus is filtered into your Bowman's capsule non-selectively except the plasma proteins. So if your concentration of glucose in your blood increases, its filtration is also going to increase. So you can see it over here in this graph that as the concentration of glucose in your plasma is increasing, your filtration of glucose is also increasing. Now let us see about the reabsorption of this glucose. Let us show this reabsorption with the blue color. So as the concentration of glucose in your blood is increasing, the filtration is increasing, as you can see over here. Now what about reabsorption? To some extent the reabsorption is going to be equal to your filtration. जितना फिल्टर होगा ग्लूकोज उतना रीअब्जॉर्ब होता रहेगा, ठीक है. सो as you are increasing the concentration of glucose in your plasma, your reabsorption is increasing as much as your filtration. जितना filter हो रहा है इतना ही reabsorb हो रहा है. And then comes a time when your reabsorption is not equal to your filtration. जितना filter हो रहा था उतना reabsorb नहीं हो रहा, and this is represented by this curve on the graph, and this is known as a splay. इसको कहते हैं splay, SPLAY. And you can see that your reabsorption is not moving along with your filtration, and some of the glucose is not, which is being filtered, is not being reabsorbed. And the concentration of glucose in plasma at which this happens is 200 milligrams per minute. So when it reaches around 200 milligram, that is when some of the glucose starts escaping into the urine, and this is known as threshold. This is known as threshold. When some of the glucose starts appearing in the urine, this is known as threshold. Now, why is threshold achieved? As not all people are the same, not all nephrons of your kidneys are same. They do not have the same abilities, and some of your nephrons are saturated earlier than other nephrons. You have 2 million nephrons in both of your kidneys. Some of these nephrons are less able to reabsorb glucose, and they have lower thresholds. So these nephrons which have lesser threshold start secreting glucose in the urine, while rest of your nephrons are still reabsorbing glucose. So because of these leaky nephrons, your reabsorption is now not equal 100% and not equal to your filtration, and this happens when glucose reaches concentration of 200 milligrams in your plasma. And when you further increase the concentration of glucose in your plasma, the reabsorption of glucose is now going to become constant, and no more further glucose will be reabsorbed, and this is depicted by a straight plateau or a straight line on your graph. And this is when all of your nephrons, all 2 million of your nephrons are saturated, and this is known as transport maximum, and this is achieved at 375 milligrams of glucose concentration. So for BDS students, this graph will not be asked of you in your exams. This was just for your understanding. What you do need to know is the definition of transport maximum and the normal tubular load of glucose, which is 125 milligrams per deciliter per minute, and then the threshold at which some of the nephrons become saturated and the glucose starts appearing in the glucose. The threshold for glucose is around 200, and then when the transport maximum of all of your nephrons is reached and no more glucose is going to be reabsorbed and all of it is going to appear in the urine, this is known as 375 milligrams per minute, and it is known as transport maximum of glucose. Then we have substances which are being transported passively, and as I mentioned before, that only actively transported substances can show transport maximum, because these substances require carrier proteins for their transport, and carrier proteins tend to become saturated, and those substances which are not being actively transported, such as passively transported substances, they do not show transport maximum. In such substances, when you increase the concentration of something, its reabsorption is going to increase as well, but they do show another kind of limitation, which is known as gradient time transport. So in a passively transported substance, the greater the gradient, the greater will be the transportation, and the greater the time the substance stays in contact with the luminal wall, the greater will be the transportation. So in passively transported substances, there is no transport maximum, and it only depends upon the gradient and the time for which the solute comes in contact with the surface of the epithelial cells, so that it can be reabsorbed. So you must remember that transport maximum is shown by actively transported substances, and passively transported substances depend upon the concentration gradient, the electrochemical gradient, and the time of contact between the substance which is to be reabsorbed and the luminal membrane. So this is known as a gradient time transport. Now coming to the reabsorption of water, please remember that reabsorption of water is coupled with the reabsorption of sodium. Greater the reabsorption of sodium, greater will be the reabsorption of water, because water is always going to follow sodium, and it is going to follow sodium either through transcellular route or through the paracellular route. But remember that all parts of the nephrons are not equally permeable to water. Some parts are have greater permeability of water, while some are less permeable, and some of them are even impermeable to water. So here I am going to draw the nephron and its different parts and show you different permeabilities of these parts. So first of all, we have the Bowman's capsule and the proximal convoluted tubule. These parts are very permeable to water. Then comes the loop of Henle. First we have the descending limb, and then we have the ascending limb. The permeability of the descending limb is also very high. It is very permeable to water, but the descending limb is absolutely impermeable to water. The ascending limb, sorry, is impermeable to water no matter what. So the ascending thin limb and the ascending thick limb of loop of Henle are absolutely impermeable of water, and water cannot be reabsorbed from these parts. Same is the case with the early part of the distal convoluted tubule. Here we have the distal convoluted tubule. It can be divided into two parts, the early part and the late part. So the early part is also impermeable to water. Now coming to the late part of the distal convoluted tubule and the collecting ducts, they are normally impermeable to water, but they become highly permeable whenever there is secretion of ADH. So in the presence of ADH, the distal convoluted tubule and your collecting tubules and collecting ducts become highly permeable to water. So this is how water is reabsorbed from different parts of the nephron. This is very important. You have to remember this. This is going to be it is going to be very helpful while studying the concepts of urine formation and concentrated urine formation, and they can be asked of you in viva and also in written exams. Don't worry, we are going to be revising them again in future. So for now you must remember that water follows sodium, and different parts of nephrons have different permeability for water. Then chloride is also absorbed along its electrical and chemical gradient. So in the nephron, most of the sodium is reabsorbed in the proximal convoluted tubule. When sodium is reabsorbed, water is also going to follow, and when there is lesser water in the nephron, the chloride which is left behind is going to be in very high concentration. So it is going to move from a higher concentration to lower concentration along its electrical chemical gradient. Also, when sodium leaves the nephron from the proximal convoluted tubule, you can see that a positive ion is being lost from the proximal convoluted tubule into the interstitial space and peritubular capillaries. There is going to be left behind an unbalanced negatively charged chloride, and this negatively charged chloride is also going to move along its electrical gradient. So chloride is reabsorbed along its electrical and chemical gradient. Let me repeat that suppose this is our Bowman's capsule and we have our proximal convoluted tubule. So in the earlier parts of the proximal convoluted tubule, a lot of sodium is going to be reabsorbed, and as you know, जहाँ sodium जाता है water also follows. Now when water leaves, the chloride which is left behind is going to become concentrated, and this chloride is going to move from higher concentration to lower concentration along its chemical gradient. Also, when the sodium left the proximal convoluted tubule, it also took away its positive charge, and now left behind is chloride with its negative negative charge, and this negatively charged chloride is going to move along the electrical gradient from negativity towards positivity. So both chemical and electrical gradients of chloride are going to push it from the proximal convoluted tubule into the interstitium and then into the peritubular capillaries. So chloride is reabsorbed along its electrical and chemical gradient. Then we have the reabsorption of urea. As water is reabsorbed, the concentration of urea in the lumen also increases, and urea is transported from the lumen back into the interstitium with the help of urea transporters. We will be discussing the reabsorption of urea in greater details in further slides in next chapter. Then we have creatinine, and creatinine is a very large molecule. Also it is a waste product, so none of the creatinine is reabsorbed from your nephron into your peritubular capillaries, and all of it is excreted in the urine. So this was a discussion of reabsorption of different substances along your nephron. I hope you understood the concepts. If you have any questions, please leave them in the comments below. Thank you.